Hello everybody, this is Dr. Vishal Trivedi from Department of Biosciences and Bioengineering, IIT Guwahati. And what we were discussing about the different chapters related to your biology course under this IIT PAL program. And in this series, let's today start the discussion about the new chapter, and the new chapter is the biotechnology principle and processes. But before getting into the detail of this particular chapter, let's discuss about the biotechnology and how the biotechnology is helping the humans or the scientists to develop the different types of molecules. So what is mean by the biotechnology? Biotechnology is a field of science where you are using the living organism or the product of living organism for the human benefits to make a product or to solve a problem. What that mean is that the biotechnology is using the technology aspects of science and utilizing that to or applying that to the living organism, or it is applying that particular technology to produce the product from the living organisms. During this process, it always benefits the human being either by bringing a new product or to solve pre-existing problems. For example, if you are producing a product from a living organisms, what the living organism is going to do is it is actually going to produce that particular product, but in addition to that, it is also producing the non-specific or the product which are actually toxic in nature. So in these cases, what the scientists are doing, they are utilizing the different aspects related to biotechnology, and that's how they are actually either diminishing the level of the toxic compound, or they are enhancing the production of that main product, and as a result the toxicity of the particular product is going to be reduced. So this is the what is mean by the availability of the new product or to the solve the pre-existing problems. So in simpler word, the biotechnology is the summation of all the activities involving technological tools and the living organism in such a way that it will enhance the efficiency of the productions, which means the biotechnology is not very limited to only the technology aspect, but also it is also helping in terms of the developing the different types of tools and other kinds of things. So biotechnology is a very vast subject, and it is actually encompassing many areas of the science, and that's how it is actually trying to understand the living organisms in a better way, and that's how it is actually trying to solve the different types of problems, or it is also uh, because once you understand the metabolism of a particular organism, you can be able to modulate the different steps what is present in these metabolic steps, and that's how you can be able to enhance as well as the make it the product make the product production more efficient. So what is the goal of the biotechnology? The ultimate goal of the field is to improve the product yield from the living organism, either by employing the principle of bioengineering or the bioprocess technology, or genetically modifying the organisms. What is mean by the biotechnology is that the biotechnology had different types of tools. One of the tool is that it can use the bioengineering or the bioprocess technology, as well as it also can use the genetically modified organisms. For example, you can have the organism which is producing a particular type of product, and that product is actually be produced at a very low rate, or it is also being produced at a very low level. So because of the principles what is being derived by the bioengineering or the bioprocess technology, you can be able to study the protein the particular product production and from the organisms, and then you try to understand the different types of factors what is governing the production, and that's how you can be able to modulate these factors, and that's how you can be able to enhance the production. Apart from that, some of the sometime what happen is the organism is deficient in some of the key enzymes, or organism is producing the product, but the product is getting accumulated within the cell, or the product is getting degraded because of some other activity of some other enzyme, or sometimes the product is being formed but it is not very stable. So in those cases, the scientists are also exploring the options of generating the genetically modified organism, which means they are actually changing the organisms in such a way that it is actually going to produce that particular product at a very large scale. But the question comes, why the biotechnology, or why the humans have started using the technology part to do all this? Because of the many problems, and one of the problem is that the human population is also increasing. So let's see how the human population is increasing over the course of time. So what you see here is a projected human population growth. So what you see is a growth of somewhere around from 1750 to 2001. And what you see here is that the human population is increasing on a logarithmic scale, and that's how it is actually increasing at a very, very rapid state. And according to a rough estimate, there will be a supply of, there will be enough food, there should be enough food to supply the projected population of 9.2 billion by the 2050, which means there will be a huge demand or on the resources, as well as there's a huge demand onto the production of different types of food grains. Now you know that the earth agricultural land is very limited. For example, in India itself, the agricultural land is reducing day by day because of the urbanizations, because of the industrializations, and all other kinds of activities. Because you have seen that the many of the roads are coming up on the highways and all other places, and from where these roads are coming, these roads are being built onto the agricultural land. So because of that, the agricultural land is reducing, and there is a requirement to produce more and more food grains, vegetables, and all other kinds of products. So because of this, there is a need to enhance the food productions from the pre-existing from the existing crops. But that is not possible, because every crop has its maximum limit until which it can actually be able to produce. So in that case, what you have to do is you have to enhance the production of the crop, either by employing the bioprocess or the biotechnology related tools, or you have to employ the genetically modified organism, which means you have to produce the crops from the genetically modified plants. And so this is one of the aspects which biotechnology is very much required, because once you have the biotechnology tools and techniques available, then you can be able to utilize that to produce the different types of crops. You can actually be able to enhance the production, either by bringing the better crops which are actually producing more number of food grains, or their production cycle is also very, very less. For example, if you have a wheat crop, and normally the wheat crops takes 4 to 5 months, but if you come up with a wheat plant which actually can do the production in 2.5 months, then you actually can do the production twice, because you can take the you can sow these particular type of wheat plants, and in 2.5 months it is going to be ripened, and then you can actually regrow the same plant again and again, and that's how you can be able to get the better production. The other aspect is that the plant can be of susceptible to some kind of infectious or some kind of insects. That also can be overcome if you are producing the genetically modified plants, and that will be resistant for these kind of degradation. So this is one aspect where the biotechnology is contributing, and that is what because is required, because there is a enhanced growth of the human populations. Apart from that, the biotechnology is also being used to produce different types of products. Uh, so, what is the need of the biotechnology? So, there are multiple ways in which the biotechnology can be able to utilize. For example, biotechnology can be used to produce the living stocks. For example, you can be able to produce the stock of the living stock, means you can have the purity of the living stock, because you know that the most of the living stocks are actually breeding to each other, and that's how they are very impure, and the impure stocks are very difficult to use in terms of the breeding purposes, because then you are actually producing the hybrid plants, hybrid living stocks, or the hybrid varieties, and these hybrid varieties you cannot control the traits. So because of that, there is a need to have the pure living stocks. Apart from that, as I think already discussed, that the some of the metabolic toxic byproducts are being secreted from these plants, and because they are producing from the plant as a byproduct, you also can be able to reduce the secretion of toxic metabolites from the plants. The third is the plants are not been able to withstand the harsh biochemical process or the treatment. So what happen is sometimes the product is being produced as the as a raw products, and then these raw products are not good to be consumed. So that's how you it actually gone to the biochemical processes or the treatment, and many of the plant as well as many of the animals could not be able to withstand these kind of harsh treatments. So because of that, there is a need to have the plant as well as the different types of treatments to be used, so that it should be able to withstand the harsh treatments. The ultimate, what we have also discussed, that there is a need to have the higher production as well as the lower cost, because once you have the higher production of the crop, eventually when the production is going to be more, it is actually going to reduce the cost of that particular crop. Biotechnology is being used very oftenly to produce the products what we consume in a daily life. For example, you are very much familiar with the curd, you are very much familiar with the bread. Biotechnology is also being used to produce the different types of wines. These wines are different in terms of the flavors, their shelf life, and all other kinds of properties. Apart from that, the biotechnology is very extensively in use to produce or to improve the quality of the products from the poultry, whether it is the weight of the chicken or weight of the egg or other kinds of properties what is being required to be from the poultry products. For example, it can actually increase the shelf life of the eggs what is being coming from the hens, or it can also increase or decrease the level of cholesterol and other kinds of fat from the eggs. And apart from that, it also can improve the quality of the hen or the chicken what is coming out from the poultry farms. Apart from that, the biotechnology is also being used to produce the different types of fishes. These fishes could be of two varieties: either it could be a ornamental fishes or to the edible fishes. In the case of edible fishes, the biotechnology is helping to produce the different types of fishes, or they are allowing the disease free fishes production, and also sometimes the biotechnology is helping to produce the fishes which are very rich in terms of the antioxidant levels or other kinds of therapeutic molecules. So if you consume these fishes, you are not only going to get the nutrients from the fish, but also you are going to get the some of the antioxidant as well as the therapeutic molecules which are actually going to make the life more healthy as well as the more sustainable. Apart from that, the biotechnology is also helping to produce the different types of ornamental fishes. These ornamental fishes where the people are actually overexpressing the different types of the fluorescent proteins. Some of the So these fishes are actually being in a high demand in other countries like European countries or the American countries, where people are actually using these fishes in the aquariums. And when there is a when they don't turn even when they don't turn on the lights, these fishes actually glow into the aquarium, and that's how the name of these fishes are known as the glow fishes. So the glow fishes are the first generation of the transgenic fishes what is being produced by the scientists, and that's how they are using these glow fishes for a very large production, and they have a very high market in the European as well as the American country. Apart from that, the biotechnology is also being used to produce different types of transgenic animals. You might have heard about the transgenic animal like Dolly, which is being produced by the scientists. And apart from that, the biotechnology is also helping to produce the different types of transgenic animals. These transgenic animals, the purpose of producing these transgenic animals is very much like what we have discussed for the fishes also, because these most of these transgenic animals are being used for the nutritional purposes. So apart from nutritional purposes, the biotechnology is also helping to produce these animals with the predefined characteristics. For example, some of the fishes, some of the transgenic animals are producing the milk, which contains very high quantity of the vitamins and all other kinds of therapeutic molecules. Apart from that, the transgenic animals are also being good in terms of the breeding purposes, and they are also good in terms of the milk productions. Apart from that, the biotechnology is also utilizing the different types of tools to produce the test tube babies. So what is mean by test tube baby is that you are actually artificially doing the fertilizations within a test tube, and then once a zygote is being developed, then you are implanting the zygote into the womb of a particular woman, and that's how you are actually helping the parents or the couples who are not having the offsprings. Apart from that, the biotechnology is also developing the different types of principles and the techniques so that you can be able to do the drug deliveries. Whether the drug delivery you are using the simple syringe, or you are using the molecular syringes, or you are using the drug delivery vehicles, all these are being developed because the biotechnology is utilizing the science part, and the scientific experiments are being performed, and that's how you are actually coming up with the policies and the procedures so that you can be able to deliver a particular drug at the site of its action. So what is the advantage of that is because then the consumption of the drugs is going to be reduced. For example, if I am getting the fever, I have to take a drug of, for example, the paracetamol of the 500 milligrams. But if I know that I am getting the fever or headache, and I just need to deliver these particular medicines only to the head part, then what I can do is I can just use the drug delivery vehicles, and I can deliver the paracetamol directly into my head area. And that's how I don't need to a 500 milligram capsule. I can just take the 100 milligrams or 50 milligrams, because then the delivery is going to be more targeted. Apart from that, so once you have the targeted delivery, it is actually going to reduce the toxicity associated with these, or the side effect associated with these medicines. Because, well, if you are taking the 500 milligrams, the 450 milligram is actually being consumed anywhere else, because, and that's how it is actually bringing the side effects as well as the toxicity, whereas the only the 50 milligram went to a actual site of action, and that's how you are actually can be able to reduce the toxicity. Apart from that, the biotechnology is extensively being used to produce the different types of vaccines, whether it is vaccines for the cholera, or whether it is a vaccine for the TB, or whether it is a vaccine for all other infectious and non-infectious diseases. You might have heard recently that the people are trying really hard to develop the vaccine against the COVID-19, because vaccine is producing and giving you the natural immunity against a particular disease, and that could all possible because you could be able to utilize the biotechnology principle, and you can be able to utilize the procedure what is being evolved during the course of action, or during the course of this particular technology, and that's how you can be able to generate the vaccines, and those vaccines are going to help the humans to fight against the infectious organisms. Apart from that, the biotechnology is also being used to generate the different types of transgenic cells. The purpose of these transgenic cells is that it helps in terms of the producing the different types of hormones or different types of biologically oriented products. For example, if you can have the transgenic pancreas and if you have the transgenic beta cells, then these cells are good in terms of producing the insulin. So if you have the insulin producing cells and if you have the scaffold where you can be able to grow these cells, you can be able to generate or you can be able to produce the artificial pancreas. And that is what people are doing when they are developing or when they are trying to utilize the tissue engineering technology, and that's how they are actually trying to develop different types of transgenic cells, and that's how they are actually implanting these cells into different types of scaffolds, and that's how they are trying to develop the artificial organs. This is going to be very helpful because it is actually going to reduce the pressure onto the donors to give the different organs. And the other advantage is because these cells are going to be derived from the humans or the person itself, there will be no chance of graft rejections. I'm sure you might have heard all these terms like graft rejections and organs and all that in your previous lectures. Apart from that, the biotechnology is also helping and trying to develop the different types of medicines or the drugs. These medicines, either it could be a medicine for simple headache or it could be a medicine for taking care of different types of infectious disease like hepatitis, dengue, and all other kind of diseases, or TB. Or it could be the medicines which are actually being for the metabolic or the non-infectious diseases like hypertension, diabetes, and all other kinds of diseases. So all this is possible because the biotechnology is helping to design the different types of drugs. Apart from that, the lastly, what we also have discussed very extensively, that the biotechnology is helping to develop the different types of transgenic crops, and all these transgenic crops are good in terms of fighting against the infectious organism like the different types of diseases. So you can have the disease resistance crops, or it can have the different types of crops which can fight against the insects. So these are insects fighting, or the insect fighting crops, or insect resistance crop. And in some cases you can also have the transgenic crops which actually can have the different types of gene being produced. So even when you are taking or when you are eating the rice, you are also getting the different types of vitamins. So that is what is being done when the people have started producing the different types of different types of rice varieties, and these rice varieties were more popular because they are not only giving you the nutrition, but they are also going to give you the different types of vitamins and all other kinds of molecules. So these are the brief overview of the different types of biotechnology products, what is being possible because we understood the principle, we understood the different types of mechanism. So before getting, so now let's discuss how the people have discovered the different types of mechanisms and how you can actually utilize that to produce the different types of products. So we take the two examples, and we take the two example of the very common example, so that you will be understand how the scientists have done the different types of experiments to understand the processes, and then how they have actually utilized that process to produce the particular type of products. So in this particular examples, we will first take the example of the bread. You know how the bread is being produced. Bread is being produced where you are actually taking a dove, and then you are actually taking a dove. So you see the dove is of a very small size. So once you take the dove and you mix up it with the yeast, what happens, it gets swelled, and then when you put it into the oven and you bake it, then you are actually going to produce the bread. And bread is being characterized by different types of the pore-like structure. Now let me ask you whether you know how the pores are being formed in the breads, or why a fermentation of the dough is actually making it more spongy and That's how it is actually being utilized to produce the different types of breads, or whether it is breads or cake or all other kinds of bakery products. Now, once the people have started discovering the metabolic processes, like how the glucose is utilized in glycolysis and Krebs cycle and pentose phosphate pathway, what they have realized is that when the glucose is being utilized, it is being utilized and produces the two major products. One is the energy. So energy is always being used by the organism for the growth of particular organism. But apart from that, as a metabolic byproduct, the glucose is, when it is getting oxidized under the different metabolic reactions, it is also producing the large quantity of CO2, and you know that the CO2 is a gas. So once the CO2 is being produced, it escapes from the dove, and that's how. So because dove is a very solid structure, so it once it escapes from the dove, it actually makes the pores, and that's how it is actually makes the whole process or whole dove very fluffy. So because the people have studied the metabolic reactions and people have studied the different types of processes, they have employed these to produce the bread. So what they done, they have taken the yeast and they have mixed up the yeast with the dough, and that's how they have incubated it for some time. So in this process, the what is yeast is doing, yeast is utilizing the sugar or the glucose what is present within the wheat or the dough, and it is majorly it is being used to produce the energy part, and that energy is being used by the yeast to make the growth. So that's why if you, even if you add the very small amount of yeast, the yeast will replicate or grow, and that's how it is actually being going to increase its number. Apart from that, the yeast is also going to produce the large quantity of carbon dioxide, and that carbon dioxide will escape from the dough, and once it escapes, it is actually going to make the tiny tunnels. Like, so you can see that if there is a, this is a yeast or the dough block, the carbon dioxide will, when the carbon dioxide will escape, it is also going to make the different types of tunnels, and that's how it is actually going to escape. So apart from these pores, it is also going to make the capillary like stuff into the whole dough, and that's how it is actually going to make the dough very spongy. So when you bake this, you actually increases this process to a very rapid speed, and that's how it is actually. Now let's move on to the next product and discuss about that, which is also being produced utilizing the different types of principles of the biotechnology. So you might have consumed this particular product in your daily life. So let's discuss about that. So the next product is the curd, and you might have consumed the curd. So how the curd is being formed? You pour the milk in a deep pan and boil it on a medium flame for 1 to 2 minutes or until the milk is lukewarm. And while stirring occasionally, you keep it on a side. Then you spread the curd evenly at the bottom of a deep bowl. So you are actually going to have the curd which is been previously been formed, and that you are going to add into the milk, which is going to be lukewarm, not very hot or not very cold. Should be medium hot, and then you cover the lid with a and keep it aside on in a warm place for 4 hours or until the curd sets, which means it could vary from 4 hours if it is a summer, or it could be overnight if it is a winter. Then you refrigerate and use as required. For example, so the curd is being formed from the milk with the help of a bacteria known as the lactobacilli species. So there are multiple species of lactobacilli which actually can be able to convert the milk into the curd. How they doing actually? The milk is also containing the sugar. So this sugar is being consumed by the bacteria. This bacteria is utilizing the different types of metabolic products or metabolic reactions, and that's how it is actually producing the energy. This energy is being used for the growth of that particular bacteria, and while the bacteria is growing, it is utilizing more and more glucose what is present in this particular milk, and as a result it is actually converting that into a lactic acid. So lactic acid is actually a metabolic byproduct of this particular bacteria. The main metabolic reactions are actually being utilized to produce the energy, whereas which is in used to produce for the growth of the bacteria, but as a side product it is actually producing the lactic acid, and lactic acid is actually reducing the pH of that particular milk. So once it is reducing the pH of the milk, it is actually bringing the milk to a pH where the milk protein are actually going to precipitate, and as a result the milk is going to be get converted into the curd. So once the people have started discussing about or started exploring the glucose and its metabolic products, or the glucose metabolism, whether it is happening in the glycolysis, Krebs cycles, pentose phosphate pathway, or the anaerobic oxidations and all of the kind of thing, what they have realized is that this particular bacterial species is very efficient in terms of producing a large quantity of lactic acid, and this lactic acid is very good in terms of bringing the pH of a milk low on a very slow speed, and because of that it is actually precipitating the milk protein, and that's how it is actually producing the curd. So, by discovering all these reactions, they started utilizing the lactobacilli species for converting the milk into the curd, and that's how they are actually utilizing this to it's because once they know all these processes, they can be able to control these processes, and that's how they can be able to produce a curd which is going to be sweet rather than sour. You might have experienced when your mom or your grandmother is producing the curd in your home. Sometimes it becomes very sweet and then you like it, but sometimes it is very sour, and that happens because the more or the less production of the lactic acid. So if there will be a less production of lactic acid, the milk is going to be precipitate, and it is still be going to have the small quantity of the sugar part, and that's how it is actually going to be sweet in taste. But if it is producing the large quantity of lactic acid, because the temperature is very high and the it has consumed all the sugar what is present in the milk, then the curd is going to be very sour. But you can imagine that if I am working in the Amul industry and if I am trying to produce the curd, it is actually what I am trying to give the curd which is going to be sweet in every time. So because of that, what I will going to do is I am going to control the multiple processes or multiple parameters, for example, and that all is being possible because I have studied the curd formation processes utilizing the different tools and techniques what is available within the biotechnology, and that's how it is actually I have produced the curd which is very sweet. And how you are doing that? You are doing that simply because you are adding the enough quantity of inoculum, which means you are adding the enough quantity of the previously produced curd, and you are also keeping it on a adequate temperature, because so that the fermentation is not going to be too much or too little, because if it is too little then you are going to have the some amount of free milk present in the process, and if it is too high it is going to give you the sour curd. So all these processes as well as the parameters are being under tight control, and that's how it is actually going to give you the sweet curd every time. And all these possible because the people have studied the multiple parameters, multiple processes what is being governing the production of curd. So if you are interested to read more about the curd making, you can actually be able to go to this particular link, and that actually is going to explain you in detail about how to produce the curd from the milk and what are the different parameters you should keep it, and what are the different parameters are going to influence the curd formations. So what we have discussed the formation of the two products. One is we have discussed about the bread, the other what we have discussed about the curd formation. And what is the learning outcome from this particular type of examples? Well, learning outcome is that once you understand a process, you can be able to modulate that particular process, and you can be able to utilize it to produce the different types of products. For example, in this case, we understood how the bread is being formed, how the yeast is utilizing the sugar what is present in the dough, and that's how we have actually come up with the idea of utilizing it into the fermentation as well as the baking process, so that you can be able to produce the bread. Similarly, we have discovered the different types of bacteria which actually, like for example, lactobacilli species bacteria, and we know that the lactobacilli species bacteria are producing the lactic acid, and that is very precipitating the milk protein, and that's how it is producing the curd. So we have utilized that also. But this is all possible because you are utilizing the pre-existing or the existing organisms. But in so many cases, what happen is that the existing organisms may not be good enough to catalyze or to perform these metabolic reactions. So in those cases, people are trying to evolve and trying to generate genetically modified organisms. So all these curd making, bread, wine, alcohol, all these was possible before the pre-genomic era. In the post-genomic era, people thought that why not we start changing the the organism itself and try to develop the organism of its choice, so that it actually going to serve the multiple purposes. For example, it can actually help the to produce the more amount of curd, but it can also help in terms of producing the some more metabolic products and so on, so that you while you are taking the while you are taking the curd, you can also get the more benefits by consuming these other products as well. So how you can do that? You can do that simply because the biotechnology relies on the two aspects. One is you are actually going to use the genetic engineering, and the other is the bioprocess engineering. So what is mean by the genetic engineering? Genetic engineering means that you are actually going to change the genome of that particular organism. And once you do the genetic changes, or once you change the genome of that particular organism, you are going to develop the genetically modified organism. And all the processes what is being involved in genetic engineering to change the genome of a particular organism are considered to be upstream processing. Similarly, once you have produced the genetically modified organisms, you can bring it into the bioprocess engineering. What is mean by bioprocess engineering is that then you can be able to study the growth and the manipulation of that particular genetically modified organism. You can be able to study the production of that particular product from these genetically modified organisms, and then you can also be able to optimize or the see how to produce the product from that particular organisms. And all these three aspects, whether the growth and the manipulation of genetic organisms, optimization of the protein product productions, or the purification of the products, are coming under the bioprocess engineering. And since the genetic engineering to produce the genetically modified organisms is known as the upstream processes, these bioprocess engineering processes are known as the downstream processing. And this downstream processing is helpful in terms of the manipulation of the genetic modified organism and the optimization as well as the purification of the products. There are multiple ways in which the genome of a particular organism can be modified. So, what is mean by the genetic engineering? The technique to alter the chemistry of genetic material, whether it is DNA or the RNA, to introduce these into the host organism and thus change the phenotype of the host organisms, are fall under the genetic engineering. Genetic engineering of a mammalian cells or a bacterial cell can be done in two ways. One, You can go with the natural mechanism, like you can actually go with the sexual reproductions. So in the sexual reproduction, you have a male which is actually going to have the blue balls, and you can have the female which is going to have the red color balls, or the genome is going to be red color. So once they go with the sexual reproductions, the 50% gene are going to be contribute from the male, the 50% gene is going to be contribute from the female, and that's how you are actually going to generate a hybrid variety, and that is going to be the progeny. So in the progeny, you have actually genetically modified the that particular organisms, but and you have generated the hybrid organisms. But the organism is hybrid, but it is not actually going to be generated as per your will. Whatever you can have the genes from the female, that is going to be random. It could be coming from this ball, or it could be coming from this ball, or it could be a mixture of both. Whereas in same is true for the male. You can be able coming from this ball, or you can be coming from this ball, or it could be coming from the both of the balls. So the DNA part is coming from the both, and you are actually contributing or altering the genetic material of the progeny, but you don't have the very specific control over what are the genetic material you want to contribute, or what are the genetic material should not contribute, because it could possible that the many of the genes from the females are coming, but they may not be contributing, or they may not be dominant enough. Because if you remember the genetic rules of the mammalian or Mendelian inheritance rules, the gene contributing a gene is not enough, it should also be dominant. So those kind of parameter actually makes the sexual reproduction as a mechanism to produce the to produce the genetically modified organism is not very successful, because of the simple reason that it actually can actually bring the disadvantage, or it can also bring the unwanted traits. Apart from that, there is the other mechanism. The other mechanism is that you introduce the DNA into a particular cell. So you imagine that you are trying to produce a particular protein which is corresponding to this particular gene. So you can just introduce this particular DNA into the cell. But if you take the DNA and introduce it into the cell has a mechanism to identify this external DNA, and that's how it is actually going to be destroyed, because you are not so because the cell has its own self defense mechanism. So because of that, it is actually going to destroy this external DNA. So how to do that? How to introduce the DNA into the cell, so that the cell should experience, or cell to realize that this is its own self DNA, and that's how it actually can utilize this DNA to produce the protein. What you have to do is you have to clone this DNA into a suitable vector, the vector which is belonging to particular host strain, which means if it is a mammalian cell, the vector could be of mammalian origin. If it is a bacterial cell, the host could be, or the vector could be of the bacterial origin. So once you clone the DNA into the vector of the host or origin, then the cell is not going to destroy this particular DNA. Then the cell will realize that it is actually its own cell, or it is actually its own DNA, and that's how it is actually going to allow the replication as well as the production of that particular protein. And then what you do is you derive the different mechanism, and that's how the DNA can be introduced into the organism. Once you introduce the DNA into the organisms, it could be reached to the two different locations. It could reach to the nucleus, or it could reach to the cytosol. If it is reaching to the nucleus and it is integrating within the genome, then you are actually going to produce the genetically modified organisms, which is going to be permanent, which means you are actually going to generate the transgenic cells, because then it will replicate, it will get self-replicated along with the genome. But if it is reaching to the cytosol, then it is going to be transient genetically modified organisms. This will stay for some time, but once the cell is going to divide, the whatever the content is present into the cytosol will not going to go along with the cell, and that's how the cloned DNA is going to be removed from the cell. So you might have to do the entry of the DNA into the organisms multiple times when if it is reaching to the cytosol. If it is reaching to the nucleus, you might have to do it once, and then you are actually going to generate the permanent transgenic cells or permanent transgenic organisms. If you want to do all these genetic modifications, you might have to go through with the multiple steps. For example, you might have to first identify the gene from the genome. So what you have to do is you have to first isolate the genome. So the procedure wise, first you have to isolate the genomic DNA. Then with the help of the polymerase chain reactions, you are going to produce the gene or the DNA what you are planning to introduce, and then with the help of the restriction digestion you are actually going to generate the sticky ends on the both sides, and then you are going to do the same procedure for the particular vector. So in this case I have taken an example of the bacterial vector, which is plasmids. So if you do the restriction digestion, that vector is also going to generate the sticky ends, and that's how it is actually going to give you the and then you are going to do the ligations. So that ligation is actually going to ligate the gene with the plasmids, and that's how you are going to get the ligated plasmids, and then you are actually going to do the process of called transformation. So transformation is actually being done in the competent cells, and then you are going to do the transformation. So transformation is actually going to do the entry of the DNA into the cell, and once the DNA is going to be entered into the cell, then you are actually going to do the screening part, and that screening is going to give you the desirable clones. Apart from the procedures, you also have to use or understand the different types of tools. For example, you have to understand about the polymerases, you might have to understand about the restriction enzymes, you have to understand about the ligases, which are actually being utilized to ligate the cut gene with the cut plasmid and give you the ligated or the chimeric DNA. And then you also should understand about the different types of the cloning vectors, whether the cloning vector for the bacteria, fungi, yeast, or the mammalian cells, or the virus-based cloning vectors. So this is all about the genetic engineering. Apart from the genetic engineering, you can also be able to do many things related to the bioprocess engineering, whether it is the growth and manipulation of the genomic genetically modified organism, optimization of the product, or the purification of the product. So within the downstream processing, you have the multiple processes what you are going to do to achieve the better production of the product from the genetically modified organism. Let's see what are those procedures. So I have already discussed about the upstream processes. Within the downstream processes, first thing what you have to do is you have to do the cell disruption. So once you break the cell, you are actually and you can use the different types of processes. You can use the chemical processes, you can use enzymatic processes, you can do mechanical processes, and you can do the physical processes to break the cells. Once the cell is broken, then it is actually going to give you the crude product, which means that it is going to give you the different types of cellular contents what is present. Then you can do the different types of extraction procedures. So because of this extraction procedure, you can be able to do the extraction of your product. You can do the concentration, extraction, and filtrations. Once you got the product, you can be able to do the concentrations. So for concentration, you can use the evaporation, ultrafiltration, or precipitations. And after this, you have to do the purifications. For the purification, you can use the chromatography as a technique. And once you are done with the chromatography, you are going to get the final product. So these downstream processings are actually going to help you in terms of recovery of that particular product what the genetically modified organism is producing. So this is all about the upstream as well as the downstream process. So this is just a brief summary of what are the processes you have to do when you are doing in the upstream processes or the downstream processes. So in our subsequent lecture, we are actually going to discuss about the genetic engineering in detail, and we are actually going to take up all the processes. So what we are going to discuss? We are going to discuss the different types of tools. For example, we are going to discuss about the polymerases, and how the polymerase is contributing in terms of the genetic engineering protocols. Then we discuss about restriction enzymes, ligases, and cloning vectors. Apart from that, we also going to discuss about the procedures. So in this particular chapter, which is related to biotechnology, we are going to discuss about the different types of tools and as well as the different types of procedures. So with this, I would like to conclude my lecture here. In our subsequent lectures, we are going to discuss about the tools as well as the procedures what is related to biotechnology. Thank you.